Oh no, not this again. Yeah, it's this again. The problem with studying anything in just the abstract, like we did here, is that it remains so abstract, it just kind of loses some meaning. I'll give you an example. What do you mean by DNA? Well, DNA. It's the chromosomes. Yeah, well, what DNA? Oh, it's the genes. Sure, but what genes? There's 20,000 genes, and they're in every single cell. So which one do you mean? I don't know, just pick one. Well, here's the point. It's not just one gene, and it's not all of them. Each cell expresses probably somewhere between 10 and 30% of the genes within it. 10 to 30% of that 20,000. That's not a lot, but it makes sense, right? That's how a brain cell and a liver cell become different, because they express different parts of the genome. That makes sense. The questions that automatically arise from this then is, well, how do we turn the other genes off? And also maybe, how do we turn on a gene when we need it, and then turn it off when we don't? Can we do those things? Well, sure. Let's take a look. All right, well, do you remember what these arrows stood for? This one was what? Transcription, and then translation. Right, you got it. So, where can we control gene expression? Transcription or translation? Well, the answer is yes. Either one. Probably makes more sense if we're going to stop gene expression to stop it here. Otherwise, it's a real waste to make RNA if we're never going to make the protein. If you're a bacterium, that's pretty much the only way you control genes, is you just don't make the RNA. You don't do transcription. We do a little bit of both, but a lot of it is here. So let's take a look at different ways we can stop, or at least control, transcription. One way to stop transcription is by coiling up the DNA. So we can really only use our DNA if it's more or less unwound like this. When we have the DNA wrapped around histones and that's coiled up into really tight coils like we find it in chromosomes, how we see it a lot of times before cell division like this, well, that's just too tight. RNA polymerase, that enzyme that does transcription, can't get in there. And so cells oftentimes coil up the DNA that they're not using, but these little puffy bits that we see here and up here, those are actually somewhat uncoiled, and that allows RNA transcription to happen. There's an interesting example of this with X chromosome inactivation. So we know that females have 2 X chromosomes, and very often one of those X chromosomes is supercoiled into a bar body that's so tightly coiled it's not used. We can actually see those in these microscope images as those little pink dots. Let's think of it this way. When we look at this case on the right of this unusual skin pigmentation condition, and let's say early on this woman had 2 X chromosomes, and as those cells divided, there's sort of this decision made. We're going to keep dad's X chromosome active here and mom's active here. So at some point there's this decision being made by the cells, which one are we going to supercoil? And when this cell divides, it keeps that decision of dad's X chromosome, and this one keeps mom's X chromosome. And the cells that result from that, more of mom's and more, and you get the idea. So all of the skin cells that result from mom's, let's just say in this case, result in this lighter color skin, and the dads result in this darker color skin, because only the skin pigmentation gene on the 1 X chromosome is actually being displayed. We usually don't see cases like this, but it's a very interesting display of it. So on a similar note of development, we all start out life as kind of chemically lopsided. And so by the time we divide and become 2 cells, those 2 cells are chemically different. Genetically the same, but chemically different. And by the time we reach 32 cells, then certain cells are signaling other cells, as we can see here, and that determines essentially what is transcribed in the nucleus of that other cell. Gene control, so cells signaling other cells. Some of these genes determine body plans. We call those Hox genes. The more general term, master genes, you'll hear sometimes, are genes that control other genes, or more specifically, the gene expression. And a lot of this work was done at Columbia University by Thomas Morgan and his lab in the early 1900s, and they determined that in fruit flies, there was these different chemical segments set up. And these determined the overall body plan, so that you would get eyes up here on the head and wings developing in the middle of the body and 3 pairs of legs and so on. You get the idea. And they did this mostly by creating mutants. Put them in an X-ray machine or give them other mutagens, and this is what a normal fruit fly head looks like. Isn't it cute? But then there are mutant forms. Some would grow legs where their antenna should be, and some would even develop some eye spots on their wings because these master genes malfunctioned and say, hey, put an eye over here or an antenna over there. And so this just shows us that even really small changes could actually have a big effect on a body plan. Wouldn't actually be that difficult for humans to grow an extra arm. Actually, one common mutation that happens is for humans to grow extra nipples. And that doesn't happen all that infrequently because we are mammals after all. And if you look at other mammals, usually they have more than 2. Well, one last bit, and a lot has been done in the last decade or so on this, which is epigenetics. And so even though we inherit our DNA and genes and alleles, and all that's well and good, and our genotype might be this, we can also overwrite those, essentially going like this. And the chemical version of that is to sometimes add chemicals onto those proteins that our DNA is wound around, the histones, or directly to the DNA itself. That's known as methylation. There are a few versions of this, but essentially those turn genes off. So you may have one genotype, but if you add a methyl group to it, then it essentially silences that. And this may last actually even a few generations. Your offspring can inherit this methylation pattern, and it seems to be caused by different environmental factors. Even unhealthy eating, smoking, things like this can cause methylation. An interesting sort of subfield of genetics that seems to be really adding a lot to our understanding of genetic diseases and disorders. Well, there's a few ways we can just outright stop transcription or kind of turn it on and off. There's another one in that sort of weird in-between stage in eukaryotes like us. Remember when we got RNA just fresh off the press and we had those little bits, those segments that were introns and exons? We had to cut out the introns and then ditch them. Well, that actually provides us the opportunity to do some weird stuff. It's called alternative splicing. So that cutting out was splicing. And normally we put this exon and this exon together and we'd call it good. But what if we did this one and then this one and then this one? We could come up with different combinations and that would make different proteins. And we do that actually in making antibodies, for example. So that's kind of another interesting semi-related note. Let's move on to translation and talk about a couple of ways we might control gene expression there. And we'd probably do this if we know that we want to make a protein within a cell, but we don't want to wait. So we want to have the RNA ready to go and not wait as long to make the protein. We don't want to start from scratch, in other words. Let's take a look at those. So one of these is microRNAs, pretty recent discovery. And so here we take our DNA in a gene and we make messenger RNA, but we can take another section of DNA and make a microRNA. And this microRNA is complementary to the messenger RNA right there. And so it makes it double-stranded RNA, which blocks that ribosome from getting into this DNA and translating it. So we don't get the protein. It makes it hold off a little bit. Some really interesting potential for medical applications here. We already take advantage of stopping transcription and translation when we use antibiotics. So they target different parts of the bacterial cell, either the cell wall or even the replication by targeting synthesis or even RNA synthesis. So some of them actually do target transcription. But the ones I want to focus on are these, the ribosomes. Some antibiotics, like the ones you see listed here, target specific parts of the ribosomes. And here we need to understand that bacterial ribosomes are different than eukaryotic ribosomes. And so this is the beauty of antibiotics, is that they harm these unique chemical features of bacteria without harming us at all. And so these are able to basically block translation, block protein synthesis, and really stymie a major part of the bacterial life process, not being able to make proteins. One potential side effect of these over here is that they can also affect our mitochondria. And can you think of why that might be? Well, this goes back to endosymbiosis theory, which said that our mitochondria are really these kind of live-in bacteria. They're sort of captured bacteria. And remember, they have a lot in common with bacteria themselves. And this is why taking certain antibiotics can harm our mitochondria, and we need to be careful about how often and how long we take those. Well, there's translation. We can even go one step beyond that and say, I've made my final protein. I have the amino acid sequence, it's been folded up, and now I have a whole protein made and ready to go, but it's actually still kind of off, right? I haven't let it go yet. We talked about this with enzyme regulation. So this is basically coming back around to the same thing, saying, I have a protein, and another way I can activate that is, for example, to add a phosphate group, which changes this active site, and now that protein is activated. So we can even wait till the very last minute, and that still kind of falls under, at least in a broad sense, this idea of gene regulation. Well, there's gene regulation. It's taken this abstract idea, this central dogma that DNA codes for RNA, which codes for proteins, and said, let's be more specific about that. Which of those 20,000 genes am I going to express in a cell? And that's going to determine the character of my cell. Is it a skin cell or a brain cell? Is it a liver cell or something lining the large intestine? All of those are going to require different genes, and our body has to have some way of controlling that. Bacteria even do this themselves. You'll learn more about their particular style when you get to microbiology later on. It's an important process, but kind of impressive that a bunch of molecules in a nucleus can somehow sort out which genes need to be expressed and which ones don't. Life is pretty amazing, isn't it?